It's an honor to be here at this meeting with the SSE and the PA. I have spoken in the past at both conferences, but my first joint meeting, and I'm going to talk about a topic today that I have talked about in the past, but it's been like at least 10 years since I presented my work on DNA and its response to subtle energies. You can see the original title, that's the official title. Then I added a subtitle called Anomalies in Subtle Energy Research. And if you stick to the very end, I have a list of all the anomalous findings that have occurred as a result of all this research. So hang in there. This is my contact information. And I want to start by giving you an outline of what I'm going to talk about. So I've done a lot of research with healers and how they affect DNA. So we'll start with what I call direct effects. That is to say, the energy emitted from these healers is directed at a vial of DNA with water and measured various properties of the DNA, whether the DNA is purified or whether the DNA is in a tumor cell. And I measured the rewinding of the purified DNA, the electrical properties, and the frequencies of the DNA. Then we're going to talk about direct effects on water, measuring the similar parameters. And then we're going to talk about how the information that's stored in the water and also crystals can then be read and interpreted by the biological system, whether it's DNA or cells. And that's the overview, and that's what I'm calling an inert carrier. And the real message of this talk is to what extent can the information and the energy associated with a healer can be stored, and how long does it stay in the various inert carriers. Okay, so I just basically have a pretty picture here that says the same thing. The direct effects are literally the energy directly on the cells or the purified DNA, and the indirect effects are either via water or via crystals. So this is a summary of the old research that I did many years ago that is worth briefly mentioning, because I was able to show that in this case, we're measuring DNA synthesis in tumor cells, and the tumor cells, the growth of the tumor cells was measured, and a healer directly affected the cells in the Petri dish, and was able to show, I was able to show that different intentions can either increase or decrease DNA synthesis. Certain intentions were more effective than others. Thought, as in mental healing, is ineffective unless there is some emotional content, particularly the feeling of love, although love by itself is not effective. And imagery and intention are the key components to making this whole thing work. So then, more recently, I've been studying the effects of healers on purified DNA, and I've done a lot of work using the winding and unwinding of the two strands with a UV spectrophotometer. But after that, I want to talk about the more recent work, which is measuring the electrical properties of DNA using a very sophisticated technique called nonlinear dielectric spectroscopy, in which case you measure the impedance, which is indirectly related to the conduction or the conductivity of the DNA. So whatever you see on the screen, just flip it in your mind and say, oh, if it's inhibiting the impedance, you're increasing the conductivity. And conductivity is kind of more sexy anyway, and popular, and people talk about the conductivity of DNA, how it's affected by the ability of DNA to heal itself. So there's a direct correlation there, which is why it's a particular interest. Then, in addition, I also measured the frequencies of DNA using the same technique. Okay, so here's an experiment that I did with different healers from different schools. And needless to say, they have different techniques. And lo and behold, when you partially unwind the DNA and then rewind it spontaneously in the presence of healing energy, most healers slow down that rate of rewinding by virtue of the slope that you could see here, going back toward very slow that it's not rewinding at all, which would be a horizontal line. And you can see the control in the middle where it says Reiki 1, should say. Oh no, in dotted line, sorry. Reiki 1 and Reiki 2 are two different practitioners. There's the control. And I've actually done that with lots of different kinds of healers, and they all slow down the rewinding of the DNA. And that actually in itself is an anomaly, because you would expect that a healer would speed up the process as the DNA returns to its natural intact helic shape. But alas, they all slow it down. Until I met Eric Pearl of the Reconnection, and we tested 3 different practitioners of that technique, and they all speed up the rewinding process, which is interesting, suggesting to me that there are at least 2 types of healing energies out there, those that speed up, at least in reference to DNA, speed up the biological system, and those that slow it down. Okay, so slowing it down, here I have tested other healers are on the right, and 2 different kinds of machines are in the middle there, and you could just showing this to reconfirm that healers are more powerful than machines, at least in terms of resonance with DNA. So that's good news. That's not an anomaly. That's what we would expect, what I would expect. Okay, so now let's talk about direct effects of bioenergy from actually 3 different healers on the electrical properties of DNA. In this case, we're measuring impedance, and you can see that some healers increase the electrical properties and some decrease it, all to varying degrees. The dotted lines, plus or minus 5%, 8% varies, is the control. So these guys are way beyond the variation you'd expect in the control, indicating that indeed energy from a healer can directly resonate with the DNA and change its electrical properties. As far as I know, no one ever had done this before. I guess you could say it's an anomaly that some healers increase it and some decrease it, because I would have thought it should increase, because again, increasing conductivity or decreasing impedance is associated with the natural healing process of DNA when it gets damaged. Okay, so now the other technique is actually measuring the frequencies themselves. This impedance spectroscopy technique allows you to measure specific frequencies, and here is a graph showing the impedance versus frequency, where you can see very discrete peaks. And here are many of the peaks in DNA, showing that this particular healer, depending on the frequency, could either decrease or increase the electrical properties at that frequency. So this is interesting, and in this case, this was done with the Qigong master in the dark bars, and the light blue bars are the control. Next slide shows the specific frequencies that a Qigong master actually turn on. You can see some of the bars go to the bottom and decrease, but I'm particularly interested in the frequencies that get turned on. So these frequencies were quite low. In fact, the 94 kilohertz frequency was not even present in the control situation. But yeah, so here we're expressing the percent change compared to the control. So these frequencies are present in DNA, although in the case of 94, barely. And the healing energy activates the DNA at that particular frequency. Okay, so the next slide is looking at the variability, or the reproducibility of this phenomena, because we have it's the same healer on two different occasions. And you can see that the frequency doesn't always appear to be activated like in the previous trial. The best example is the two in the middle there, where you could see that it's close. The frequency is not exactly the same, but similar, which indicates that the frequencies are not discrete and concrete. They actually fluctuate. When you measure them, they actually hop. And this phenomenon called frequency hopping is in the scientific literature. So you don't always get the frequency at 94 kilohertz. It fluctuates, and depending on the treatment, as you can see in this the second treatment didn't affect the 94 kilohertz signal at all. So there's a lot of variability here, and I guess you could call that an anomaly, because I can't really explain that. Okay, so here is two different healers affecting the DNA, showing this is basically the same conclusion that we saw from the previous graph, just expressed differently. Okay, so now let's talk about the ability of inert carriers to store this information. And we're going to look at three different kinds of carriers: liquid water, or bulk water, and liquid crystalline water, which is another fancy way of saying structured water, and crystals, like as in hard physical crystals. And I have done experiments with different healers on different types of waters and different kinds of crystals, which you are about to hear. So the background for this is the idea of imprinting water with healing energy. Was actually probably first done by Schwartz, and he measured changes in infrared, and then Bill Tiller came along, measured changes in pH, and then I did a lot of experiments using ultraviolet spectroscopy. But there's actually a lot of studies which demonstrate the physical, chemical, and electrical properties of water change in response to a healer. Okay, that's nice, but does that mean that the information is imprinted? And the way we can answer that question is to see whether or not a biological system can read that information. So it's more than just changing the properties of water, it's storing the information in the water itself. So here's an example of 4 different healers, where in this case, the effects was on the electric part of the water. Oh, so before I talk about treating the water itself, this is the effects. No, I'm sorry, this is water, for the property of water. Okay, sorry, we're in water. I have to keep moving my video camera here so I can see the title. So, okay, so they're treating the water, and then you add the water to the DNA with the information, and the dotted bars are the controls. So they do vary, obviously, plus and minus 8, 9, 10%. And the healers, in all cases, decrease the impedance or increase the conductivity of the DNA in response to the energy that's stored in the water. Okay, so the conclusion from these kind of studies is that yes, water information can be imprinted in water, and that water remembers this information, and that biological systems can read the information. Okay, this was actually first published in 1995. I mean, there's scientific rationale for doing all these experiments. A bioelectromagnetics researcher asked the question, well, all cells respond to electromagnetic fields, but all cells are surrounded by water. What's the role of water in the response of a cell to electromagnetic fields? And they did the brilliant experiment that no one ever done before, and just zapped the water with the electromagnetic field and added the water to the cells, and they got a biological effect, as you can see. In this case, they're measuring the electrical properties of the potassium channel. So anyway, so my experiments were done with tumor cells grown in tissue culture, where water was treated with different intentions, and that water was used to make tissue culture medium, and then measured the effects on cell growth of the tumors. And here we're comparing direct and indirect. So in the direct experiment, the healer zapped the Petri dish with the tumor cells, and the indirect experiment, they zapped the water, which was then used to make the culture medium. And as you can see, the direct effect is somewhat stronger. Not surprising. No anomaly there. About here, however, we have the indirect effect being much larger than the direct effect, which was an anomaly for sure, because it's not the way it's supposed to be. So that's an interesting point for this group particularly. Okay, so now if we look at indirect effects from different kinds of water. Okay, so you can see that when distilled water, DI, it was what I had used previously, but when you add salt to that distilled water, you produce the opposite effect. Tap water itself had no effect, and spring water, which does have minerals in it, some sodium, but not as high concentration as in saline, you back to the original effect with distilled water. So this is very much dependent on the mineral content of the water. That's not really surprising. So then the question was, well, let's look at structured water. Now there's a commercial company that I work with called Vivo. Here's the Vivo water, which is structured water confirmed by NMR spectroscopy, lasts for 2 years, published data, patented data. There's a lot of research on Vivo water. So here we use Vivo water to receive the energy from a healer, and it didn't work. Distilled water had the predictable effect. The unstructured mineral solution, UMS, had the opposite effect of distilled water, but then that's because of the presence of all the minerals. So that's not surprising. What's surprising is that structured water did not take on the information from a healer. But if you think about it, the water is so structured and so organized, it's not open to receive information. So it doesn't imprint. Interesting observation. Okay, so now we're going to move on to crystals. Same experiment, you treat the crystals, and then in this case, you put the water sample on top of the crystal in a glass container, so the energy is emitted from the crystals, goes through the glass, through the water, and affects the DNA. And you can see that different kinds of crystals have different kinds of effects. Again, lapis and tiger's eye have the opposite effects, and some are weak and some are strong. So, I mean, that's interesting by itself. Yeah, here we're asking the question, how long does this phenomena last? And if you treat the crystals with bioenergy with 2 different intentions, you could see that certainly with intention number 2, by the time you get to the 2nd month, the information is almost back to 0, which is actually a really long time. And the anomalous effect here is that some intentions, it doesn't decay, it gets stronger over time. So that's an anomaly right there. Okay, so then we do the same experiment, this time not with a energy from a person, but with a scalar energy from a caduceus coil. And this time, the information in the water decays over the course of several days, not several months. So this is encouraging that water imprinted with healing energy lasts a lot longer than man-made energy. I suppose that's not really an anomaly. Okay, so now we're moving on to experiments here that Bill Tiller talked about. He did the same kind of experiment measuring pH, and you can see here, he gets very symmetrical, coherent changes in the pH over time. And now I want to talk about yet another way of storing information. So Bill Tiller stored his information not in a crystal, not in water, but in this kind of electronic circuit. That's the main point, which was rather novel at the first of its kind. No one had even thought of doing that. but he was able to get reproducible results, and he published it in the journal Scientific Exploration. So hats off to the SSE for publishing this, because it was so far ahead of its time. Nowadays, the latest is to try to store information in a wave file in a computer, and this information is then transferred to the water, like we've been talking about, and you generate what's called an infoceutical. And so instead of a nutraceutical, which has the chemical or the herb or the drug, it's an infoceutical, which has the information or the energy of the drug in the water. And these two websites are two existing companies. The first one's in Russia and the second one's in Europe, that actually sell these infoceutical products. But in the scientific quest for knowledge, we have moved on to a company called the Chi-Box that I've been working with, who has patented technology for recording and storing the bioenergy of a healer using special scalar antennas. The information is then stored in a wave file on a small Android device, and the Android devices transmit the information to regenerate the information directly into the body, or as you'll see, into water. Each device has several different intentions, and the company already has some preliminary clinical evidence for efficacy in regards to detoxification. They don't have a website yet, it's such a new company. But what they also have is these recordings of what the energy looks like. But before I show you that, I wanted to show you the effects of two different programs, one that treats allergies and one for treating Lyme. And you might think, well, what is and again, we're measuring the electrical properties of DNA. What does a DNA have to do with allergies? Well, you can see on the bottom of the screen an article published in 2002 directly linking those two phenomena. So there is justification, not an anomaly. You would expect the DNA to respond. But here's the kind of recorded spectra they get from the wave file. Once you have the information in a wave file, it's there forever. So there's no issue about how long it lasts. You just play back the wave file. So here's a recording from a healer before they were putting out active energy and when they were. So you can see on the axis on the right is frequency, and the axis on the left, if you will, this is the right and the left. And I described that. The right is frequency, and on the left is time. So you've got. Oh, I can use my cursor here. So you've got time, 0, time, low frequency, high frequency. These are all actually very low frequencies. Here's a recording from a healer, and this is, I'm sorry, this is the recording and the direct measurement, which indicates that when they measure the healer directly, the energy coming from the healer and the energy coming from the recording, they're pretty much the same, except you can see one little difference around there, but otherwise it's quite similar. Okay, so here's two different programs, which two different intentions, which you can see are actually quite different. And again, these peaks represent, the activity at a particular frequency at a particular time. Different crystals emit different kinds of patterns. If you look over here, this is the Marcel Vogel crystal that people who know Marcel Vogel's work is quite remarkable, because not only does it have the low frequencies, which are different than the other two, it's also got the very, very high frequencies. So quite remarkable crystal. So getting toward the end here, just looking now at the effects in summary when comparing direct a healer directly on either the lymphocyte growth or DNA versus the indirect effect when the healer treats the water and then the water is put on the biological system. And the point to make here is that when you use an electromagnetic field, actually this is a scalar field, the indirect effect is somewhat less. In this particular experiment, it was recorded with an audio recording. And this experiment, which is, again, significantly less, but this is the G-box results, which are actually quite similar. And this particular method of storing the energy appears to work the best. So in the interest of time, I think I've covered all the anomalies throughout the course of the lecture. No one's giving me a hook, so I don't want to, it's been 25 minutes, so you guess you can read this to yourself. But to me, there are a lot of interesting findings as a result of this research. And if I had more time, I could elaborate more time. I don't have time for the methodology, which is why I put it in the end. But here's what happens for the unwinding when you unwind the two strands. These are the kind of control curves you get as a function of time. This is the electrical properties of DNA using this nonlinear dielectric spectroscopy. And these are the kind of electrical measurements that you can make. And in the interest of time, I'm just going to thank everybody for your patience, and I'll be available for questions and answers so we can go into the methodology if you really want. Thanks very much. Been a pleasure.